You set to go? No. Come on, Nicole. Weekends. You were supposed to have recovered last week from fall break. Oh, PCAM. Oh, I thought you said weekends. Nice going. Oh.
I never did. I was not complaining.
Okay, Wednesday lab. There's a fair amount of reading to do prior to lab. If you want to know what you're going to be doing, save you time if you know what you're doing. You're going to be gathering a lot of information about your unknown. We will be inoculating some agar plates, and those agar plates will contain various foods in their environment, like protein or nucleic acid or lipid. And we're going to see if your organism has the ability to produce and secrete enzymes out into the environment, which will digest those foods and allow them to be used in metabolism. Then we're also going to look at the fermentation of various carbohydrates. So we'll do some fermentation broth tests. We'll do a fairly complicated test called the triple sugar iron agar test. We'll do a litmus milk test. If it comes in time, we'll do an OMPG test. Otherwise, we'll wait on that. So a fair amount of inoculation. So make sure that you have a good deal of growth on a fairly recent slant for lab on Wednesday. It doesn't have to be a particular age, but it's got to be viable, and you've got to have enough that you can inoculate several different kinds of media. Okay? So last week we were talking about the tissues of the body and said that we would concentrate our discussions on 2 of those 4 tissues. We talked about epithelial tissue a little bit and the difference between keratinized epithelium and epithelial mucosa and some of the adaptations that those tissues had to allow them to be preventative barriers against infection. I also want to say a couple of words about connective tissue, because connective tissues can be important too. What characterizes connective tissue? Yeah, it's a bunch of living cells that are oftentimes not in direct contact with one another, like they are epithelial tissue, but they're embedded in a non-living matrix. What are some examples of connective tissues in your body? Bone, cartilage, tendons, ligaments, and blood. Okay, so there's really 2 kinds of tissue. There's what we call typical connective tissue, which tends to bind and support things. So that's the bone, the cartilage, tendons, etc. And those cells are usually embedded in some kind of a solid matrix. Okay? If it's bone, it's a mixture of calcium phosphate salts. What's a protein that's very important in the matrix of connective tissue? Collagen. Yep. Collagen's a very important connective tissue. Blood we sometimes call atypical connective tissue because the matrix is not solid, it is liquid. And then what are the principal cell types we find in the liquid that make up that blood connective tissue? Platelets are one type. What are their 2 principal blood cells? Red blood cells, erythrocytes, and white blood cells, which are leukocytes. Okay? So we'll say a few words about connective tissue as we talk about invasion by microorganisms, and then blood will become a very important of our discussion when we talk about the body's defense systems. Okay, we introduced the concept last time of virulence. What do we mean when we say something is virulent? Huh?
It is able to infect.
Yeah, it's able to infect and cause disease, okay? Because remember, those 2 things aren't the same. Infection is just multiplication within the tissues, and then disease is a manifestation that may result from that multiplication. It may not, okay? So when we talk about virulence, there were also different levels of virulence. Some organisms were highly virulent, and those that really don't cause disease but do infect and don't produce symptoms are said to be avirulent. But if you cause infection, you've got to be able to breach those barriers that normally prevent infection. And one of the things that typically allows you to breach those barriers are a variety of things called virulence factors. So these are things that help to make microorganisms more disease-causing, if you like. And when we start going through some of these and some of these organisms, you'll see that some of the most infective agents have a really, really impressive battery of virulence factors. They don't just have one, they have multiple of these things that make them really highly invasive. Okay, we tend to group virulence factors into three categories. One category are the exoenzymes. The second category are the toxins. And we typically break toxins up into two categories, endotoxins and exotoxins. We'll talk about that later on. And the third category are some anti-phagocytic factors. So that's what this slide up here is showing you, is those three particular categories of virulence factors and some examples which we will expand on and talk about in a little bit more detail. In some respects, you can think of these virulence factors as the offensive weapons that microorganisms possess that allow them to overwhelm your body's natural defense systems. Many of these compounds are biochemicals, and particularly many of them are proteins. As these biochemicals start to accumulate in your tissues, they have an effect on cells. And there are consequences of those effects, many times cell death. The type of substance that kills the cell, the type of cell that's killed, and where that cell is located, oftentimes produce characteristic symptoms of that particular infective agent. So when a doctor is trying to diagnose disease, they oftentimes are looking for particular symptoms that give you some insight into what is the causative agent. And we know from prior experience that it's really important to know what the causative agent is if you're going to treat it properly. Okay? At a very fundamental level, for example, it's very important to know whether the disease is caused by a virus or bacteria, because viruses don't respond to antibiotics. Okay? So many of these biochemicals are responsible for the symptoms associated with the disease. So what we're going to do is we're going to run through some of these virulence factors, and then where appropriate, we're going to talk about some of the organisms that produce them, some of the diseases that result, that you may have become familiar with just in your general understanding. Okay? So let's start out at the top and talk about exoenzymes. Obviously, if these are enzymes, these are proteins. Sometimes these particular proteins are not encoded by the bacterium's genome, and instead the bacteria have to acquire the genes from another source before they can produce these particular enzymes. For example, there are some virus that can infect bacteria, and when the virus infects the bacteria, it donates some genes to that bacterial cell. and it doesn't kill it. It actually incorporates those viral genes into the bacterial chromosome. Does anybody know what that state is called when a bacterial cell, or a human cell for that matter, can harbor foreign genes from a virus without the cell being killed? I don't know if you remember from cell biology or if Dr. Dunn talked about that, but there are two life cycles for a virus. There's a lytic cycle where when the cell is infected by the virus, it causes the cell to burst and release more virus. But there's also a type of life cycle called the lysogenic life cycle. And that's where the viral nucleic acid is incorporated into the cell's chromosome. And it lies there kind of dormant. It replicates every time the cell's nucleic acid replicates. But there can be consequences. And in one particular case we're going to talk about, one of the consequences is the bacteria can now produce a toxin, or an exoenzyme that can produce. Okay? What does the word exo infer? Okay, these are secreted enzymes. Okay? They're produced by the cell and then have an action outside of the cell. There are several exoenzymes that have the ability to interact with epithelial tissue. So if a virus infects you or even contacts you, if it can produce some of these exoenzymes, then that ability might compromise the barrier of the skin and allow that bacteria to invade. What are some examples of some of those exoenzymes? An example is an enzyme called hyaluronidase. The substrate for hyaluronidase is a complex compound called hyaluronic acid. And the thing you need to know about hyaluronic acid is that it makes up kind of an intracellular cement that holds cells together within a tissue. Okay, remember we said our definition of a tissue was a group of cells that were connected to one another? What keeps those cells connected? Well, there are some intercellular junctions, for example, but then there's this sticky stuff between cells that helps to hold those cells together. And that's really important in places like the lumen of the intestine. Okay? You've got all these epithelial cells, and they make this tube that runs down your body. Okay? What's the function of those epithelial intestinal cells? Absorption. Okay, and do they want to absorb just anything? No, they want to be selective, and so there are various ion pumps and membrane carriers that pull some things out of the lumen of the intestine and into your blood, but leave other things behind. Okay? Would it be a good idea if substances could move between the cell junctions of adjacent epithelial cells? No, that would defeat the purpose of having those selective membranes that filter things out. Okay? So having an intact intestinal lumen is really important. Okay? Well, if the bacteria can produce this hyaluronidase, cells start falling apart. And if they are skin cells, then that gives you a route of entry into the lower tissues of the body. And so this can be a very important invasion mechanism. This enzyme literally starts causing cells to kind of fall apart, literally. And sometimes this is referred to as spreading factor. Another exoenzyme is keratinase. That degrades keratin. Remember, that's one of the principal proteins in epithelial tissue. tough and soluble protein that helps make those cells a good barrier. Mucinase. We know what mucin would break down? Genesis? Mucus, yep. This helps to degrade mucus, and remember mucus is beneficial in that it coats pathogens, helps to prevent them from binding to surfaces of the intestine or the lungs, for example, and makes them easier to expel. Collagenase. Breaks down collagen, breaks down the connective tissue. And finally, an enzyme called lecithinase. Lecithin has another name that you might be more familiar with. Another name for lecithin is phosphatidylcholine. Anybody know what kind of a molecule phosphatidylcholine is? It is a phospholipid. Where are phospholipids important? Cell membranes. Okay, so this degrades a principal component of cell membranes. So what's it going to do to the cells? It's going to cause cell lysis. And what's kind of interesting is when cells start lysing, not only does the cell membrane lyse, but organelle membranes lyse. And if that organelle membrane happens to be a lysosomal membrane, then now all of a sudden these digestive enzymes, which were contained within the lysosome, start spilling out and killing other cells as well. So lecithinase can cause cell death, but then also can promote adjacent cells dying as well. And that can be really important later on, and it will be, when we talk about pathogens that are anaerobes. Because in areas where cell death occurs, those pockets quickly become anaerobic, and then anaerobic spores can germinate there. So this can be important in the spread of some of those anaerobic infections. So here's a epithelial cell that is part of some tissue and probably, let's say, the lumen of the intestine. And these particular bacterial cells start cranking out these exoenzymes that start breaking down things like hyaluronic acid. Down here is some protective collagen that's being destroyed. And so that really helps that pathogen to invade and cross some of those physical barriers. Anybody know what this is? I don't know if you can probably read the little legend up here. This is gas gangrene. Okay? This is a nasty condition. Tissue becomes blackened. In gas gangrene, the tissue swells, has a very foul odor. Actually, there is more than one type of gangrene. Sometimes differentiated into the categories of wet and dry gangrene. The wet gangrene is the gas gangrene. We'll talk about that in a minute. Both of these conditions can cause tissue death, tissue necrosis, and can be spreading and progressive. Both of them are initiated when a condition of ischemia develops. You know what ischemia means? You have a condition of ischemia when blood flow gets blocked to tissues. Well, obviously, the consequence of blood flow is cell death. Blocked blood flow, cell death. In dry gangrene, there can be a number of things that can cause tissue to become ischemic. One of them is tissue trauma. It might sever blood vessels that supply that particular tissue. It might crush blood vessels which supply particular tissue. There's another thing that can lead to ischemia that is an environmental factor that can affect particularly the peripheral structures of the body, like the hands and feet. It's that environmental factor. Temperature, okay? What's your body's response to cooling of body temperature? Actually, vasoconstriction, especially of the peripheral arteries, veins, capillaries. What's the body trying to do? It is trying to divert that blood to the core. Maintaining the core body temperature is much more important, at least for a while, in trying to maintain life of the organism than getting blood supply to the extremities. So the tissues can start to become ischemic. They start to die. And then cell death can occur. As cells die and rupture, lysosomes can be released, more tissue can die. So dry gangrene can occur as a result of frostbite, severe frostbite. Okay. Wet gangrene also involves ischemia. There's got to be an initial trauma, usually, to tissue that prevents blood flow, kills cells. And as a result, that tissue quickly becomes anaerobic. But something else has to happen in order for wet gangrene to occur. And that is the wound has to be contaminated. By spores of an anaerobe bacteria called Clostridium perfringens. If the tissue was alive and aerobic, those spores wouldn't germinate because they are obligate anaerobes. But in this now damaged anaerobic environment where the tissue's been destroyed, blood flow's been disrupted, those spores can start to germinate. And all kinds of nasty things start to happen when those spores start to germinate. Clostridium perfringens can produce 11 different exotoxins. And they do all kinds of nasty things. We won't talk about all of them. But, for example, one of the toxins is that lespinase enzyme. So cells start to die. They release lysosomes. That then causes adjacent cells to die. Their lysosomes release enzymes that cause adjacent cells to die. And so the spreading begins to occur. And as tissue becomes newly killed, that anaerobic pocket extends, and then more spores can germinate. The bacteria can thrive.
What is it that's causing the adjacent cells to die?
Typically, one of the big things is that when the toxins like lecithinase kill cells, then they burst their lysosomes, and then the enzymes in the lysosome can kill other cells. Okay. These organisms also produce collagenase, so tissues kind of start to fall apart. That connective tissue, particularly the muscles, gets degraded. And the bacteria then begin to ferment muscle proteins. And one of the things that's produced during protein fermentation is a lot of CO2. So gas starts to accumulate in the tissue. That causes a swelling of the tissue. What's that going to do to blood flow? That's going to restrict blood flow even more. Okay? And so this condition tends to be progressive, 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 and eventually can be fatal. So, what are our options for treatment? Okay, in severe cases, amputation. Okay? And you have to take some healthy tissue usually as well as some infected tissue because you can't always tell whether some damage has been done and tissue might look kind of healthy, but it's already been compromised. Okay? If it is not quite as severe, then sometimes wound debridement, just really cleaning everything out really well, and then antibiotics can be indicated. Okay. There is another potential treatment that's sometimes used. Hyperbaric oxygen. What does hyperbaric refer to? High pressure, okay? So people are put in a chamber and literally oxygen is forced under pressure to infiltrate the tissues, and what does oxygen do? It'll generate some of those reactive oxygen species, and remember these anaerobes don't have the necessary enzymes to detoxify those things. Some of you have had contact with Dr. Dana before. I think Dr. Dana has used hyperbaric oxygen treatment in some of his wound care protocols. Yeah?
We saw a patient last week that had gangrene. They had the diabetes cut off.
Yeah.
But they, we didn't see what the PA did. He said that it smelled so horrible. When we walked in,
Now, do you know, was it gas gangrene or was it like a diabetes situation where the?
oh, they said it was gas.
Okay.
But it was
Yeah.
like, I don't know how they knew. I don't know if they did like a CT scan or something. I don't know how we found out.
Yeah. Yeah, I mean, I think a good diagnostician can really look and tell the difference between ischemia due to poor circulation due to diabetes, for example. I mean, a gas gangrene, there's visible swelling, and the smell is really a big tip-off too. You don't get that quite as much in the dry gangrene. If gangrene isn't treated pretty fast, mortality rate exceeds about 40%, so it's a pretty serious situation. It's not something you want to mess around with. There are some different kinds of exoenzymes that don't react with epithelial tissue, but instead react with blood tissue. One set of exoenzymes that's important in pathogens are a group of enzymes called leukocidins. Yes, ma'am?
Can you remind me why people with diabetes have poor blood flow?
I'm not really sure what all of the physiology is, why high blood sugar affects those endothelial cells, but it has something to do with the effects of the high blood sugar on the circulatory system. Yeah. Has Dr. Rhodes talked about that in physiology? What actually the high blood sugar is doing to impair circulation?
Glucose toxicity, but I don't know.
Glucose toxicity, okay.
Be the swelling response or anything.
Yeah, okay. What does a leukocytin do? Kills white blood cells. Okay. Bacteria that can kill white blood cells are oftentimes referred to as pyogenic bacteria. Pyogenic bacteria are bacteria that stimulate the formation of pus, pus-forming bacteria. So what is pus? It's an accumulation of dead white blood cells, fluids, killed pathogens. Okay? So obviously, if you produce an enzyme that can destroy an important part of the body's defense system, the white blood cells, that makes you more invasive. Another exoenzyme that reacts with blood components is an enzyme called coagulase. Coagulase causes a precipitation of some of the blood cell proteins. And those blood cell proteins can actually then precipitate on the surface of the pathogen, forming a little kind of shell of blood protein around the pathogen. Why might that be advantageous to the pathogen? Huh?
Because then the body or white blood cells can't tell the difference between the pathogen and the proteins.
Okay, it might help to disrupt the detection of that, and it certainly also interferes with the phagocytosis of that. So this is kind of like the artificial version of what structure?
Body.
Or bacterial structure? Or bacterial structure? Where Audrey was sitting? Lab exam?
Capsule.
Capsule. Okay, it's kind of creating an artificial capsule for yourself as well. Another type of exoenzyme that bacteria can produce are some kinases, and Streptococcus produces the enzyme streptokinase. Streptokinase breaks up blood clots. So it actually has a medical application for the treatment of blocked arteries in stroke. Okay, if somebody is suffering from a stroke or a potential heart attack, on board they can try to give them some aspirin immediately, which tends to inhibit some of the clotting and clot formation, so things don't get worse, hopefully. But then if you can administer streptokinase, this is a clot-busting enzyme that actually starts to break down the clot and hopefully can then restore circulation. Why would it be an advantage to a bacterium to produce this clot-busting enzyme?
It's able to injure the cells more easily.
Eventually, yeah. Well, I mean, what's the purpose of a clot? It seals ruptured vessels. Okay, so there's a very complicated clotting mechanism that is initiated by injury, where some soluble blood proteins, like plasmin, become converted into various insoluble proteins, which then form a net and basically can wall off and adhere to the ruptured vessel and start to seal it off. But what's another function of clots? Not really block blood flow, but cage up pathogens, okay? Pathogens can become trapped in this clot, and it helps to restrict them from moving around and infecting new tissue. Okay? So we're going to see that when we talk about inflammation, some of the early aspects of inflammation result to try to promote increased clotting and to help localize and keep pathogens where they might have invaded the body in the first place. So what do these kinases do? They allow the bacteria to break out of these clots. Okay? Another exoenzyme that some bacteria can produce are hemolysins. And you've seen this. Hemolysins can be easily detected by growing microorganisms on blood agar. hemolysin that is most obvious and easy to detect is the beta hemolysin, which not only kills the red blood cells, but breaks them down and degrades their hemoglobin. Why in the world would it be an advantage for a microorganism to break red blood cells and break down their hemoglobin? Possible. It might be a step in creating an anaerobic environment.
You can use the amino acids.
Possibly. You could use the amino acids that are liberated when you start to break down the hemoglobin. Hemoglobin is a protein, but it also contains something besides amino acids. What does it contain? Iron. Okay. Iron, as it turns out, is a very valuable resource for microbial growth. And it oftentimes is a limiting factor in microbial growth. We'll talk about that a little bit more later on. But actually, with some pathogenic organisms, there's kind of a tug of war that occurs between the host and the pathogen trying to get iron to support microbial growth. And some bacteria have evolved these enzymes that kind of bypass that tug of war, and they go, to heck with it. I'm just going to lyse some red blood cells and break down the hemoglobin and release the iron. Okay? Then I've got the iron that I need for growth. Second category of virulence factors are the toxins. And I'll elaborate on this a little bit more later on, but toxins are broken down into two categories: exotoxins and endotoxins. What does a toxin do?
Disrupts slow processes. I mean,
Yeah, it kills cells. Okay, these are substances that kill cells. Okay. Exotoxins are secreted proteins. They can be secreted by some species of both gram-positive and gram-negative bacteria. Basically, you've got to have the right genes to produce a toxin. Sometimes those genes are viral genes rather than bacterial genes. Sometimes the bacteria needs to be lysogenized first. Endotoxins are part of the LPS layer of a bacteria. Very specifically, they are a molecule called lipid A, which is one of the molecules in the LPS layer. So these toxins are actually a physical part of the structure of the bacteria. And they're released when a gram, positive or negative, what is LPS layer? When a gram negative bacteria dies. And then these particular toxins have some effect on cells. So what we're going to do first is we're going to look at some exotoxins and talk about a few diseases that are the result of exotoxin production by some pathogens. And then we'll come back later and talk a little bit about endotoxins. Toxins tend to be target cell specific. And oftentimes the toxin is categorized depending upon the type of cell that it tends to target. So there are hemotoxins, which affect red blood cells. There are neurotoxins, which affect nerve cells. There are enterotoxins, which affect GI tract cells. Toxins are oftentimes spread throughout the body. So they can be produced in one place, but then migrate to different parts of the body. If the toxin is spread throughout the blood, then we have a condition called toxemia. If the toxin is ingested, it is called an intoxication. A lot of the endotoxins that cause food poisonings are intoxications. But so is ingested ethanol. Okay? High enough concentration of ethanol kills cells. So what are the consequences of exotoxin production and binding of that particular toxin to a cell? Depends upon what the nature of the toxin is, and it depends upon what the cell is that it binds to. So we're going to run through a few pretty well-known exotoxins and talk about some diseases associated with these particular exotoxins. The diphtheria toxin. This is produced by a bacterium called Corynebacteria diphtheriae. Won't have any occasion to encounter this particular one in the lab. This particular species of bacteria can't produce a toxin itself. It had to have been infected by a virus, and that particular toxin is actually encoded for by viral genes that have become inserted into the bacterial cell chromosome, i.e. lysogeny. This is a communicable disease, meaning it can be transferred from person to person. So these bacteria can be spread primarily by respiratory droplets, and skin-to-skin transfer. So if you pick up one of these bacteria, pick up enough of them, depending upon the infectious dose, this has got a fairly high infectious dose, then those bacteria can start multiplying and producing that particular exotoxin. This targets epithelial cells, and very specifically, the toxin inhibits protein synthesis. It blocks an elongation factor and interferes with it. Cells can't synthesize protein, they die. One of the first places that you start to see some of the effects of diphtheria typically are in the throat. And one of the early signs of diphtheria is some generalized inflammation and a very sore throat. But as those cells in your upper respiratory tract start to die, then some necrosis can develop. We get this mass of dead tissue. One of the ways that the body responds to this particular infection is it steps up mucus production. As your immune system starts to respond, white blood cells start moving to that site of infection, and those white blood cells can be killed because their protein synthesis is inhibited. So you can form this nasty mass of dead tissue, dead white blood cells, pathogens, and that can form a structure called a pseudomembrane. And sometimes, it's a little bit hard to see without light on it, that pseudomembrane can become so thick and so extensive that it literally blocks off the respiratory passage, and your trachea becomes blocked. So you could die from suffocation. Well, what's the way you could potentially mitigate that? Yeah, either cut that out or do an emergency tracheostomy. Create a new artificial airway. Antibiotics are not that great of a treatment because it's really the toxin that is causing the problems. Yeah, the bacteria are producing the toxins, but killing the bacteria, the toxin still might be hanging around. So one of the things that really is very useful in treatment of many of these diseases are antitoxins. Antitoxins are basically antibodies that have been produced by challenging an animal, like a horse, for example, against the diphtheria toxin. Yes, the horse will die, but they can then harvest the antibodies from that horse's blood that have been generated against that particular foreign protein. And then if you administer those antibodies, the antitoxin, to an affected human, it can really help to make that person survive long enough that their immune system kicks in as well and treats the problem. Anybody ever seen the Disney movie about Baldo? What's it called? Do you remember, Hope? Yeah. Is it called Baldo? Okay. Anybody know the story? Baldo was a sled dog, and in 1925, in Alaska, there was an outbreak of diphtheria in Nome, I believe. And the antitoxin was in Anchorage, hundreds of miles away. And so they took a sled team led by Baldo that delivered the diphtheria toxin to the town and saved the people. That is actually honored now yearly by a big Alaskan event. You know what that is? The Iditarod. Okay, the annual sled dog race from Anchorage to Nome. Okay. What would be a better strategy to try to prevent getting this disease than receiving antitoxin after you've gotten it? How about being immunized against it? Okay. Do we want to give young kids active diphtheria toxin? No. They might not survive. So what we do is we treat the toxin in a number of different ways to weaken or attenuate it. Sometimes that's a heat treatment. Mildly just denatures the protein, destroys its toxicity, but it still is recognized by the immune system. Have you had diphtheria immunization? No. You have tetanus, diphtheria, and pertussis, which is whooping cough, are all part of the same vaccine treatment. Okay? Well, we'll continue to talk about some more toxins and diseases on Tuesday.